Uh, Hi, we'll start as soon as a few other people come.
Hello?
Yeah, hi. So we'll start and if in a few minutes after a few other people
Yeah,
come.
sure, sir.
Yeah, thanks. Yeah, hi Surender. I think we can start.
Yeah, sure.
Okay, so uh, now we'll be discussing a few assignment problems for week 3 of the NPTEL course, Metabolic Engineering. Uh, yeah, can you see my screen?
Yes, yes.
Okay.
So before that, can we discuss
Last week's
what
the
was
doubt, the last week
assignment?
doubt questions.
Yeah, yeah.
Okay. So yeah, so last week we had discussed on the previous week's assignment part. So one of the questions that was raised is uh, when solutes are being transported across an antiport, then can it be classified as energetically coupled or not? That was one question that was put
Mm-hmm.
across. So I did look at how antiports work, and technically the answer is yes, because as long
Okay.
as we have one of the in antiport specifically there is movement of two solutes, usually in the opposite direction. As long as one of the solute is able to move, and as long as that requires energy, if the other solute is also present in the other end of the barrier, then it can also move. So technically, it it requires energy. It is energy coupled.
Okay.
And another question that you had asked is whether the example, whether is if there is an example of active transport without carriers.
Mhm.
So I checked about this, and there are no active transport without carriers. Active transport necessitates that a carrier is or should be present for uh, transport of the molecule when energy is utilized. So I think both of it goes hand in hand. You need a carrier protein and you need energy for the solute to be transferred. And
So question number 6, it needs to be corrected, in assignment 0.
assignment 0 is not counted for. So
Uh,
maybe we can. I remember seeing your email in the thread
mhm.
in the Google groups. So they can make changes if needed.
Okay.
Yeah. Uh,
I have
yeah.
one more doubt.
Yeah. Sure.
From last week assignment, assignment 2, question
The
number 10.
questions that we discussed or the question that was a part of the actual assignment?
Part of the actual assignment.
1 minute. I'll open that.
Uh-huh.
week 2, question number 6, is it?
10.
10.
No, 10.
Okay. Huh. So the question is, in the fermentation process, organic carbon acts as electron source. Okay. Oxidative phosphorylation serves as a platform for energy production. Okay. Both oxygen and NAD plus act as electron acceptors. Okay. Concentration of NADH plus H plus increases. Which of the statements are true about fermentation process?
Mhm.
Okay. Now they are saying that the answer is B, which is options 1 and 3 are correct. So organic carbon acts as electron source. Okay, that is right because that from we have studied that carbon enters, catabolism happens, you have small metabolites, anabolism happens, you have uh, polymers that's being created, but in the absence of oxygen, certain processes are restricted. And apart from that, in most of these processes, oxygen oxygen is the end electron carrier, so it accepts electron. And so your doubt here is
Mine doubt is in fermentation, both oxygen and ND act as electron acceptors. Okay,
okay, okay,
it seems incorrect for me.
okay. You are saying in
But
fermentation, oxygen is very limited, so it can't act as an electron acceptor. If that is what
moreover,
you're saying, is it?
NAD, yeah, yeah, NADH is the electron donor here, not acceptor. And acceptor is the, for example, pyruvate may be the acceptor, electron acceptor.
I will check with them and I'll report that. Question 10,
10.
assignment 2. Okay, I will check with the team and I'll see if see what they have to say.
Okay. In the same assignment, question number 5.
5, right.
So, building
So the
materials.
question is precursor metabolites used for production of building blocks of cell wall in E. coli is option A glucose 6 phosphate, then fructose 6 phosphate, ribose 5 phosphate, and pyruvate. They are saying that the answer is B. And you are saying none of this is the part of the cell wall, is it?
No, I'm saying it may be glucose 6-phosphate because cell wall made up of peptidoglycan and peptidoglycan made up of N-acetylglucosamine and N-acetylmuramic acid. And
Yeah.
both are derived from the glucose
Hmm.
6-phosphate. That's why I have I'm little bit doubt here.
Okay. Okay. Question for assignment two. Sure, sure. I'll just check and I'll update.
Okay.
Huh. Okay. Huh. Other than this, if you have any other questions, we can even, if you want, we can
Uh,
check that. Or
what do you mean bow tie structure? What is that bow tie structure?
so in one of these lectures, if you remember, there is this reduction. How do I say? I'll maybe even open the lecture. I have the lecture number. Give me a minute. Uh, Hi, Meera Manjari Sahu. You can introduce yourself while others join. Surendra, give me a minute. I'll just check the lecture number for this bow tie structure and we'll see that.
Mhm.
I think it's lecture number 6, where they say that its entry of sugars and other carbon sources leads to precursor metabolites formation that leads to macromolecules. So I think it is what they try to say is the bow is larger than the tie with respect to the number of metabolites, maybe. So maybe the cell can take multiple carbon sources but convert them
Mhm.
into a limited number of metabolites. So that is like a
Okay.
funnel arrangement. So you have many carbon sources, but independent of those carbon sources, we get limited number of 12 precursor metabolites that directly contribute to the biomass. So the size is this much initially, and then it's getting reduced. I think that is what they wanted to mean by the bow tie structure.
Okay. Okay.
Huh.
Got it.
Okay.
I got it.
Okay.
And
So
now we
yeah,
can start to today's.
right. So yeah, so for week 3, uh, yeah, we'll start with the questions. Which of the following points are most appropriate for describing systems biology? I hope my screen is visible. Uh,
It is.
Option A, modeling of complex biological systems. Option B, an interdisciplinary field that combines the knowledge and techniques from biology, biochemistry, genomics, bioinformatics, chemistry, and computational science. Option C, it seeks to identify how biological functions evolve and how emergent properties in cells and communities arise from seemingly simple genetic sequences. And option D, all of the above. You can take a couple of minutes and then we can start discussing.
For me, all of the above is the correct.
Huh. So you can try to start with one point. We'll discuss each of the points and then so that the recorded lecture is also being useful for others. Uh, yeah, so we know that in systems biology, they try to model biological systems using mathematics. So option A is right. And because the data that they take here is assimilated from different fields, including biology, biochemistry, genomics, bioinformatics, and other stuff, option B
Mm.
is also right. But once they take the data, then they try to see if there is any patterns that tries to explain any properties within the cells or the communities. And this they do from majorly from modeling using genome scale modeling. That is what option C tries to say, which is uh, It seeks to identify how biological functions evolve and how emergent properties in cells and communities arise from seemingly simple genetic sequences. This is also right. So the answer that all of the above is right. Yeah. And the next question is two-dimensional annotations associated with option A, traditional genome annotation, option B, spatial organization of cells, option C, genome evolution and plasticity, option D, interaction of biological components. Huh. We can just take a couple of minutes and then we can discuss this. Yeah.
So the interaction.
Yeah. So in biological data sets, the kind of annotations that they do is first is the ORF based annotation that they do, but that is just limited because when you do the sequence analysis and you locate specific genes and assign functions to the genes, you know that such a gene exists in the organism. But when it comes to two-dimensional annotation, apart from assigning this function to the gene, they also try to see if any of the genes or the gene products interact with each other depending on the kind of function they perform. So, as you said, the answer is interaction of biological components. The third question is the process of finding the relative location of genes on a chromosome is called option A gene tracking, option B genome warping, option C genome mapping, option D chromosome walking.
It is mapping.
Genome mapping, yeah.
Map.
Uh, so, do you know what is the difference between genome walking or chromosome walking and genome mapping?
Genome mapping is just we have to find out the location of genes on the chromosomes with respect to each other.
Huh,
Their proper arrangement on the chromosomes.
huh.
Okay, and chromosome walking is one of the method of means to find out the complete sequence of the genome. I'm not sure, but chromosome walking is used in which we sequence the large fragments of DNA,
Hmm.
then find the overlapping regions.
Hmm. So,
Okay,
what? Yeah, sorry.
Mhm. and from this overlapping, if we find the overlapping fragments, then we can find the complete sequence from one end to the another end of the genome.
Huh. So, what is done in genome walking or chromosome walking is you already know some information about a particular region within the chromosome or the genome, and so you design complementary sequences to that and try to amplify from that region. So, that will act as a primer. So, when you do this amplification, you get to know what is the region that is flanking the region that you know. So this way you will be able to sequence from the region that you are aware of. This way, you'll be able to get the sequence of the entire genome in certain cases, like you had mentioned earlier. Yeah, so the answer is genome mapping. Huh. 1 minute, I'll accept someone is entering. Huh. Yeah, so Kritika, we've started discussing. We are in the fourth question. Once we finish all the set of questions, we'll go back and start discussing from the first for the questions that you had missed. That should be fine, right? Okay. So, Surinder, we need not tell out the right answer. We'll take chances and discuss amongst ourselves. Like Kritika is also there.
But
So, huh, yeah. So, the fourth question is, say the correct option for denoting the structural hierarchy of a metabolic network. So, we have different parts, or the metabolic network structural hierarchy can be defined into different parts, including modules, like small scale modules, large scale modules, having different components, the genotype and the phenotype. So, Kritika, you can take a couple of minutes and try to define uh, or try to answer this question as to what you think is the right answer and what is the wrong answer. Yeah, Kritika, do you want to give this a try? Oh, okay. Uh, if that's the case, then uh, Surinder, we can
mhm.
start discussing. So, yeah. So, you can share what you think is the right answer. Then we'll discuss how this goes.
Okay. Okay.
Sorry, can you come again?
It's first of all phenotype. B. I think B is the correct answer here.
B is the correct answer. That's what you think. Okay. Okay. So, okay, we'll discuss what metabolic networks are, and then we'll try to think if B is the right answer. So, a metabolic network is basically having information on the genes that are involved in a particular process, how they are connected, and the order in which they are connected. Correct. So,
Mhm.
to be able to know this information, we need to know what are the genes that are involved in the process. So, that means we don't start with a specific uh, trait that is being observed. Rather, we start with the sequences. So, if you talk about the structural hierarchy of a metabolic network, then genotype cannot be the first component. Rather, it would be sorry. So, phenotype cannot be the first component. We start with basic sequences when we are constructing metabolic networks. So, from the sequences, we do the annotation and we get an information about the genotype. Correct. So That because of that reason, phenotype cannot be in the first place. And once we get the genotype based on annotation, we get them into different components like DNA, proteins, protein coding regions, and stuff like that. These are the components. Once we get that, we try to arrange them based on the kind of flow the path the metabolic network has and the kind of interactions it has with other small different similar modules. So the let's say there is a particular metabolic network consisting of three steps, then the genes involved in that particular step alone will be called as the small scale module. But if we have some other module which encompasses this small scale module and many other small scale modules that in total contributes to a specific phenotype, then the all of the sum of modules will be called as the large scale module. So you'll have small metabolic networks that contribute to the final metabolic network, which contributes to the phenotype. So that way, if you look at it, the structural hierarchy of a metabolic network starts with the genotype, builds components from the genotypes, gets small scale modules. These small scale modules are
Com
arranged. Huh, sorry.
proteins, huh? Components means proteins. Product.
Components could be proteins or DNA or even regulatory elements. So
Okay. And
from the sequence, once you assign the ORF and the function, when you try to do the interaction, you try to see if you don't always stop yourself from with proteins alone. If you have enough information, you can also try to incorporate the interactions. That's what we were talking about in question number two, the 2D annotations. Like you had mentioned, interaction of biological components. So the components here could need not be necessarily strictly proteins alone. It could be others as well. Proteins and protein interacting components. It could even be small molecules, ligands that interact with proteins. So these form
okay.
some small scale modules, and there could be many such small scale modules that lead to the phenotype. So when these small scale modules are connected, then the network becomes large. It's called the large scale module, which then finally contributes to the phenotype. I hope that was clear.
Yeah, yeah.
Yeah. Huh. The next question is mention the type of the below represented reactions map. Options are open, close, all of the above, and none of the above. 1 minute. Kritika, we'll just discuss why option A is correct. Uh,
I think she's in metro. That's why she's not speaking.
yeah, yeah, she had put a message there. Yeah,
Mm-hmm.
we can discuss that. That this should be fine. Yeah. You can share your thoughts whether you think if it's open an open network or a closed network or if it's a combination of both. You can share. Yeah.
So, open reactions are those where the materials can be exchanged
Yeah.
with the environment.
Yeah. Sure.
So here the A is taken up by the from the environment
And
and E is going into the environment. Okay.
yeah. Yeah.
So that's why it is a open system.
Yeah. Yeah. You're right. You had explained that well. Good. Huh. We'll move on to the next question. So choose the correct sequence of biological network inside a cell. So we have three different kind of net or the metabolic or the networks within a cell can be biological networks within a cell can be divided into three types: metabolic, regulatory, and signaling networks. Um, Now we have to choose the correct sequence of the biological network. 1 minute. Kritika says it's option B. We'll take 2 minutes and then we'll start discussing this. Kritika, is it possible for you to write and explain if you can't open your mic? Yeah, that should be helpful. We'll wait for a couple of minutes, Surender. We'll see if Kritika's answer appears in the text box. If not, we'll
Mhm.
start discussing.
Okay.
Yeah. Okay, Kritika, I'll explain the answer. Uh, so, Surendra, do you have an answer in your mind which you think is right? The options are option A, metabolic network, followed by regulatory network, and followed by signaling network. Option B, signaling network, followed by regulatory network, followed by metabolic network. Option C, regulatory network, followed by signaling network, followed by metabolic network. And option D is metabolic network, followed by signaling network, followed by regulatory network.
So it's B.
Yeah, so do you have an explanation for that as to,
Because
yeah, sorry.
uh, some signals are required for any metabolic pathway to happen. Okay. For example, if in the medium
Hmm,
lactose is there,
right,
then uh, lactopron start operating. It
right,
start to synthesize the necessary enzymes
right,
that are required for the degradation of lactose.
right,
And if still if glucose plus lactose is there, then lac operon will not be active, no? So there's some regulation.
Hmm.
Okay,
Hmm.
so I think it first signaling comes, then regulation of the signaling.
Right.
Then it leads to the metabolic networks ultimately. Different kinds of the pathway they have occur, and it leads to the metabolic network.
Right. Yeah. Just building on your answer. Yeah. Option B is right. As you mentioned, the first component of any pathway involves signaling. The signal is could be either intracellular or extracellular. Let's say there is a metabolite A that is entering the cell. It could be that way, or maybe the cell requires a metabolite to be synthesized. Both of these needs signals. So either the presence of the metabolite or the absence of the metabolite acts as the signal. And this signal is linked to the signaling network is linked to the regulatory network. So the regulatory network is the one that determines whether gene expression or gene expression, which is gene upregulation or gene downregulation, should happen. Like you mentioned in the case of lac operon, what happens is when lactose enters the cell, it goes and binds to a regulatory molecule called LAC-I, which is the LAC repressor. So this
Mhm.
sits on the LAC operator and it prevents transcription of genes downstream. So this is how it is placed. You have LAC-I, then LAC promoter, then LAC operator, then LAC-Z, LAC-Y, and LAC-A. This is the arrangement within the in the genes. This is how the ORF is presented. So the LAC-I is first synthesized. It is always present on the operator. Then when lactose is available, this lactose binds to LAC operator. So LAC operator can no more bind to the DNA, which is the LAC operator region. So now the RNA polymerase can transcribe. So now here, if you when it transcribes, it produces LAC-Z, LAC-Y, and LAC-A, of which each of it has one function. One is a permease, and the other is has another function. So here, specifically in this case, like you mentioned, lactose will be the signal. The regulatory network involves lactose binding to LAC-I, and the metabolic network involves further processing of lactose. Yeah, I hope that was clear.
Yeah, yeah.
Yeah. The next question is genome scale metabolic model reconstruction depends on option A, genome annotation, option B, biochemistry data, option C, cell physiology data, option D, all of the above. We can just wait for a couple of minutes and then we can define how the components that are shown here, one or two or three, all of the components are specifically important for the reconstruction. We can just take a couple of minutes. Hi, Muskan. Uh, am I audible to you?
Yes, sir.
Yeah. So you can introduce yourself. We have other students here as well.
Good evening, everyone. I am Muskan Rahman from Mumbai. I am doing metabolic engineering from NPTL as an elective subject in my college. Thank you.
Yeah. So we started discussing. We are more than halfway through the discussion. Once we complete the remaining questions, we can again go back and check the questions that you had missed in the initial half. I hope that's fine.
Yes, sir. Sure.
Yeah. So the question that we are right now looking at is genome scale metabolic model reconstruction depends upon option A, genome annotation, option B, biochemistry data, option C, cell physiology data, and option D, all of the above. So you can take a couple of minutes, or you can try putting forward your answer as to what you think is the right answer with an explanation. We'll discuss it further as you come up with the answer. Yeah, Muskan, if you have an answer in your mind, you can share that here.
Actually, sir, there is some network issue, not able to see your screen.
Oh, Uh, others, do you have any issues? Surendra or Kritika?
No, I can see.
Okay. Okay, Muskan, if that's the case, you can have your mic on. I can read out the question. You can just try to think and let us know what you think is the right answer. Should that be fine?
Sir, let me join other. Wait.
Huh. Okay, let's go ahead with the discussion. Surrender or Kritika, you can try to mention what you think is the right answer and why specifically.
So genome scale metabolic model.
Yeah.
So, as name indicates, there should be involvement of genome.
Huh.
And metabolic means there should be biochemistry.
Right.
So in physiology and biochemistry, they are same. So
And they're not necessarily the same, but yeah.
yeah, yeah.
Huh. I'll explain it further. So you're saying all the
So
three
all
are right, is it?
yeah, yeah.
Okay. Yeah, like you said, okay, first we'll talk about what genome scale metabolic model reconstruction is, and then I'll explain why different sets of data are needed. So genome scale metabolic model reconstruction, It is basically you are able to sequence the genome of a particular organism. And once you're able to sequence it, you annotate it, meaning you identify what all genes are there and try to assign a function based on the genes that are present. And you try to give a metabolic network of sorts based on this information. So this is basically the minimal version of a genome scale model. So when you do this genome annotation, like we had discussed, it gives an idea about what genes are present, and the biochemistry data based on functional annotation tries to give you what all possible pathways can exist within the cell. The third is case specific. It is called as a cell physiology data, meaning at a particular condition, environmental condition, internally, this is what these are the genes that are being expressed or repressed or being active or inactive. That is the cell physiology data. Not all genome scale metabolic models will have cell physiology data, but that is also being included in case specific manners. So that way, the answer is all of the above. Huh. So yes, Surendra.
So in most of the cases, genome scale metabolic model reconstruction involve the gene annotation and biochemistry.
Yeah,
That
majorly because
mean,
cell physiology data is usually case specific. Although that is also being used, but uh, it's majorly the genome annotation and the cell physiology data. I mean the
but
biochemistry data, not cell physiology. Sorry. Huh. The next question is, which is the correct biochemical reaction for the given stoichiometric network, stoichiometric matrix? So here they have a matrix called S, and you have something of the sorts of Kritik. Sorry, Surendra. So Kritik has mentioned something in the chat. I'll just check that. He's mentioned answer for the last question was D. So we'll look at it. Yeah, the option is answer D. All the three are, which is genome annotation, biochemistry data, and cell physiology data. All the three are required. But in most cases, we try doing simulations with the genome annotation and the biochemistry data itself. Cell physiology is case specific. At a particular environmental condition, the cell behaves a particular way. You get an idea about something known as the flux for a particular reaction. So, but that is not always available. Yeah. So now the question that we're dealing with is the stoichiometric matrix, and we have to write the biochemical reaction for this. So this matrix is called S matrix, and in we have one column and four rows. So the column here represents the you can call that as the rate of the reaction or the flux through the reaction, whereas the rows represents the metabolites. So here the metabolites are glucose, ATP, glucose 6-phosphate, and ADP. And you have values such as -1, 1, and 1. So you can take a couple of minutes and try to tell us what the right answer is, we'll uh, based on which we'll discuss. Yeah, Kartika, we'll discuss in a couple of minutes. Yeah. Yeah, so the values here that are being shown have two different signs, positive sign and negative sign. Generally, in these stoichiometric matrix, these are reconstructed from the reactions, chemical reactions. If in a chemical reaction, if a metabolite is being consumed, then depending on the stoichiometry, that is number of moles of the metabolite that's being consumed, it is given negative and the number of moles. In this case, 1 mole of glucose and 1 mole of ATP interacts and gives 1 mole of glucose 6 phosphate and 1 mole of ADP. Because glucose and ATP are consumed in per mole basis as 1 mole, we get minus 1 and minus 1 here. Whereas glucose 6 phosphate and ADP, because they are produced as 1 mole each, we get plus 1 and plus 1 here. So the right answer is option A.
Can you explain again why minus one plus
Yeah,
one
sure,
plus one?
sure, 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 sure. So these stoichiometric matrices are usually constructed from the reactions. Okay. So in the reaction, we have something called as reactant and something called as the product. So reactants are usually consumed during the reaction and products are being produced. So to be able to represent that in a matrix format, we represent metabolites that are being consumed in negative terms and metabolites that are being produced in positive terms. And the value here, 1, this depends on the stoichiometry within the reaction. Let's say there is 1 glucose and 2 ATP, then it will not be minus 1 and minus 1, rather it will be minus 1 and minus 2. Rather it will be minus 1 and minus 2. So that is how we decide whether the values are, what the values are and what the sign is. So in any reaction, the products that are being produced are given positive sign and the reactants that are being consumed are given the negative sign. Was that clear?
Okay.
Huh.
So reactants given negative and product given positive.
Yeah, yeah, yeah. Huh. So, okay.
And what this VI stands for?
So, usually in stoichiometric matrices, like every other matrix, we have a row and column. In stoichiometric matrix, the rows represent the metabolites and the columns represent the reactions. In a metabolic network, we'll have many reactions. So we'll try to represent the entire set of reactions in one matrix in this form. Here, this is V1. This is just stating that this is reaction 1. When it comes to actual application, this V1 is given a value called as the rate or the flux. It is similar to the concept as kinetics because there also we talk about stoichiometry. True. Surinder, was it clear? Why?
Yeah, yeah.
Huh, yeah. So the next question is EC number is assigned to option A DNA, option B RNA, option C enzymes, and option D none of the above. We can just take a couple of minutes and then discuss what EC is and take it forward. Yeah, Kartika, we'll just take a couple of minutes and then decide. Yeah, Kritika. So, like you had mentioned, EC is enzyme commission number. This is assigned based on the kind of reaction an enzyme performs. So, enzymes can be categorized into seven or eight major categories based on the function that they do, like hydrolysis and other such enzymes. Uh, there is a website called Brenda. Uh, One minute, I can even present that here. Yeah, so this is Brenda. If we type an enzyme name, then we'll get that uh, enzyme's EC number. So these are the different kinds of enzymes based on the function that I had mentioned, like hydrolysis, transferases, oxidoreductases. So if we go to one of these, we have based on the kind of donors and acceptors and many other different uh, categories, we have subclasses. So this is how they are being classified, and this is applicable only for enzymes. So the right answer is option C, enzymes. Uh, then the next question is, name the relationship we give to the Boolean link between genes and reactions in the genome scale metabolic model. Option A, reaction translation gene. Option B, gene translation reaction. Option C, reaction protein gene. Option D, gene protein reaction. We can just take a couple of minutes and then start answering. Yeah, Kritika. The answer is D, gene protein reaction. Like we had uh, discussed earlier, first from the genome sequence, we get annotations for genes. Then we try to get their functions, which leads to us annotating between protein coding regions, forming the proteins that are involved in the biochemistry of the reaction that forms the reaction itself. So the relationship is called gene protein reaction. Now we'll go back to initial questions since we are done with the set of questions to be discussed for this week. Huh. So the first question is, which of the following points are most appropriate for describing systems biology? Option A, modeling of complex biological systems. Option B, an interdisciplinary field that combines the knowledge and techniques from biology, biochemistry, genomics, um, bioinformatics, chemistry, and computational science. Option C, it seeks to identify how biological functions evolve and how emergent properties in cells and communities arise from seemingly simple genetic sequences. And option D, all of the above. So, Kritika, you can take a couple of minutes and try answering this question, then we'll discuss it further. Kritika, you can type out the option in case if. Okay, so you're saying for this question also it's D. Okay. Uh, yeah. So in systems biology, we deal with modeling biological systems, um, and because it uses information from different disciplines like biology, biochemistry, genomics, bioinformatics, chemistry, computational science, and others. It's an interdisciplinary field that combines this knowledge. And apart from this, in systems biology, we start from the sequence level, trying to give annotations and then further functions, and then use information from the above mentioned disciplines. That way, we try to see how functions evolve. And currently in systems biology, apart from single cell modeling, uh, even tissue level modeling has been progressed, which is mentioned here as emergent properties in the communities. So the right answer is option D, all of the above. The next question is two-dimensional annotations are associated with option A, traditional genome annotation, option B, spatial organization of cells, option C, genome evolution, option D, interaction of biological components. Um, um, yeah, Kritika. So you can just take a couple of minutes. Okay, you have answered already, which is option D. Ha. So with respect to biological data, the traditional 1D annotation was just annotating the genome, meaning uh, based on analysis of sequences, one could able to locate the open reading frame and then assign the function based on what the gene corresponds to, if there is a gene product or not. This is the 1D genome annotation. The 2D genome annotation is, apart from assigning a function to a gene, you also try to identify if there could be any interactions between genes that have similar or common functions. So here we try to identify interactions, which is interaction of biological components. Option D. The question option C is the process of the question 3 is the process of finding the relative location of genes on a chromosome is called option A gene tracking, option B genome walking, option C genome mapping, and option D chromosome walking. And Kritika, you're saying the option is genome mapping. So do you know the difference between genome walking or chromosome walking and genome mapping, Kritika? Okay, I'll explain what the difference is so that it's helpful. So in genome mapping, what people try to identify is relative location of genes. They have a gene at a specific loci or locus. Compared to this locus, how far or how near other genes are. That is called relative location. Whereas in genome walking, what people do is they know the existing sequence within a particular region of the genome. Then they design a primer that overlaps with this region and try to amplify from the primer. So what specifically happens is, because you're amplifying, you're able to get the sequence of the gene region which you are not aware based on the region that is close to this particular gene. So that is called as genome walking. Was it clear, Kritika? Okay. Huh. The next question is, we were talking about the structural hierarchy in a metabolic network, and we were discussing what contributes to, or how is the structural hierarchy in a metabolic network given there are many different contributing links like genotype, component, different modules, phenotype. So, Kritika, you can try to answer this question and then we can. Okay, you want the answer of the last question. Okay. So, the right answer for this question is genome mapping, like you had mentioned. Huh. Uh, yeah, Kritika. I hope the screen is visible. You can try to see, uh, try to answer this question. Okay, you're saying it is option A because we start with the genome sequence. So there is some truth to what you're saying, but it's not exactly right. It is true that we start with genome sequences, but what we need to be aware is that from the genome sequence we get the genotype. So from this we know the ORFs and we assign functions. So when we assign function, we are able to restrict the length and say this region to this region is involved in a particular function that forms the components. But only when we look at specific interactions, we'll be able to come up with modules. This component and this component interacts this way. That gives the small scale modules. And only when small scale modules are assembled together, you get the large scale module. So the right option is not option A, but rather it's option D. Uh, was it clear? Yeah, okay. Huh. The next question is mention the type of the below represented reactions map. Uh, yeah, so what you see here is a reaction map having a few reactions with a box in between and a few metabolites. Some metabolites are entering and some metabolites are exiting. Okay, you have attempted these questions. Fine. So, with this, we come to the end of discussion for this week's problem questions. But because we have about half an hour's time, I thought maybe we can discuss the solved assignment uh, questions for week one and week two for this year. And if we have any doubts, we can go through them. Yeah, so we'll start with week one's assignment. Yeah, I hope my screen is visible. Huh. So we'll go with week one's assignment. So the first question is, among the various genetic modifications, which one of the following is not considered as a part of pathway manipulation through genetic engineering? The options are modifying the promoter strength of a given gene. Option B is gene deletion. Option C is introduction of new genes to the pathway. Option D is mutation followed by selective screening. So one of you can try attempting and telling telling us what you think is the right answer and why.
So genetic engineering is not random,
Hmm.
uh, and here we are targeting a particular metabolic pathway or particular gene.
Hmm.
So it is not by chance. Huh? That's
Hmm.
why.
True. Like you had mentioned, in genetic engineering, we targeted specific genes for specific reasons. Either we know that structurally a gene behaves a protein behaves a particular way, so we make changes at the gene level so that it gets reflected at the protein level. We don't do random mutagenesis and do selective screening. So although that was earlier considered, but now it is not considered as a part of the genetic modifications for pathway manipulation. The second question is, which one of the following best describes the heterologous expression of a gene? So the answers are expression of a gene or a part of gene in host organism that naturally have the same gene or fragment of the same gene. Option B, expression of a gene or part of gene in host organisms that naturally do not have the same gene or fragment of the same gene. Option C, both A and B on environmental conditions. So this is the question that we had also discussed as a part of our weekly assignment solving session. So uh, we had already discussed that it's called heterologous only when the host does not naturally have the same gene or the fragment. Question C is question 3 is which of the following steps of metabolic engineering includes the important parameters that defines the physiological state of engineering component, elucidates the control architecture of a metabolic network. So there are three options: analysis step and the synthesis step. So if we look at lecture 2, they talk about they talk about three different generations where they talk about metabolic engineering and how it has evolved. In the first generation, they just grow microbes and extract whatever it produces naturally. The second generation, they talk about this mutation and screening, which we had earlier discussed about, where the mutagenesis is random and the genetic and the metabolic profiles of the mutant strains are not very well characterized. The third generation, there is precise modification of enzymatic reactions through genetic engineering. Here, two different processes happens, or two different steps. You can classify it into two different steps. One is the analysis and one is the synthesis. So in the synthesis step, what we do is once the components are engineered, they are allowed to be expressed within the cell, and the products of our interest are getting synthesized, and that is taken care of. In the analysis step, we try to see when we change a particular component a particular way, how it specifically alters the profile in the reaction. So corresponding to that, the answer here is analysis step. Uh, the next question is four basic steps followed in metabolic engineering are design, build, test, and learn. The build step is associated with option A, metabolic flux analysis. Option B, high throughput sequencing. Option C, pathway selection. Option D, assembling genetic parts. Uh, so here, design means we try to analyze pathways or proteins involved in the pathways and try to make modifications that will might help us. So this is mostly done prior to actually working with the components. The part where we work with the components is the build step, and the test step is when you assemble all the components, put all of them together, and see if there is an improvement in the yield in metabolic engineering. Then you try to go back and see if particular set of modifications have contributed to a particular yield. What or how does it actually change? That is the learn step. So corresponding to that, the right answer here is assembling genetic parts. That is the build step. The next the next question is which step in metabolic engineering assesses the biochemical impact of genetic and enzymatic modifications for designing the further alterations to be made in the upcoming pathways? Analysis step, synthesis step, random mutagenesis. Depending on the nature of the new metabolite, one should choose the step at the start with. Uh, yeah, so like we had discussed, uh, the third generation of metabolic engineering involves two steps, which is synthesis step and the analysis step. In the synthesis step, we make the changes, and in the analysis step, we try to assess whether a particular alteration or modification would be beneficial or how it is beneficial. So, corresponding to it, here the answer is analysis step. The next question is microbial phenotypic and physiological characteristics are important for metabolic characterization of an existing or novel strain. Uh, the answer is true because unless we know how a particular strain behaves given a particular condition and what are all the phenotypes it exhibits, we will not know whether it is particularly suited for our metabolic engineering goal. So it's always better to know the microbial phenotype and the physiological nature of the strain. The next question is E. coli has been widely used as a microbial cell factory because of the following factors. Option A, high growth rate and high cell density. Option B, wide availability of gene-based models and genome engineering tools. Option C, biopharmaceuticals are largely derived from E. coli cells. Option D, all of the above. So the right answer for this question is all of the above. We had also discussed this as a part of the problem solving session 2 weeks back. Um, Question 8. A paradigm shift in the concept of metabolic processes within the living system refers to option A, individual enzyme-centric reactions, individual gene-centric reactions, interconnected biochemical reactions within the cell, genome-centric analysis. So, when we're talking about this paradigm shift in metabolic processes, what has actually helped in metabolic engineering is understanding how biochemical reactions are connected with each other, such that if you change one particular node, or in this case, one particular product or reactant in a particular way, then it doesn't affect that particular subpathway alone, it affects the entire cell's physiology. So that was able, we were able to capture that using different informations, including biochemical information. And once all of these were annotated, we were then able to generalize this once the genome sequence alone was available. So that is the paradigm shift that was discussed in metabolic engineering. Question 9. The key factors in the cost effective implementation of metabolic engineering involves option A, improve it to the point of commercial success. Option B, stitching of genes together to attain a compatible metabolic process. Option C, consider individual enzyme driven reactions. Option D, none of the above. So, the goal of metabolic engineering itself is to be able to synthesize a particular molecule of interest at a large scale in the industry, meaning it is improve it to the point of commercial process. So that is the right answer. Hmm, in question 10, which of the following holds true for computer-aided synthesis of biochemical pathways? Which is B nice. Uh, the options are it considers the chemical structure of the substrates and products. It has a non-generalized algorithm. It does not consider the enzyme rules. De novo design of enzymatic pathways. And option D, A and T. We had discussed this in week one's interactive problem solving session, where we had listed out what all Be Nice considers and what it doesn't consider. So basically, it considers the chemical structure of the substrates and products. It has a generalized algorithm. And because it knows which enzyme acts on which reactants to give which products, it also considers the enzyme rules. And based on this, de novo design of enzymatic pathways is also available. So if one is asked what is true, then it is option E. With this, we come to the end of discussion for week one's assignment. Next, we'll go through week two's assignment. Yeah, so now we'll be discussing week two's assignment problems. Identify the source or sources of precursor molecules required for the synthesis of macromolecules. Option A, glucose. Option B, other carbon sources. Option C, amino acid. And option D, all of the above. So, because they're asking about source or sources of precursor molecules required for synthesis of macromolecules, first we look at what precursor molecules are and what macromolecules are. So, precursor molecules are one among the 12 different metabolites, small molecules, that are being produced within the cell when uh, sugars are being taken up from media. These sugars could mean glucose and other carbon sources. Whereas macromolecules are the polymers like DNA, RNA, and proteins that are essential for the survival of the cell. And specifically, amino acids are required for synthesis of proteins. Since you're asking for sources of precursor molecules that are required for macromolecule synthesis, we need to consider all the four options. So the right answer is all of the above. Question two: in what pattern is the dogma of metabolism organized? So we had a discussion earlier in this session. So the right answer is bow tie pattern. What is observed is the cell can take up many different kind of sugars, but it doesn't make different metabolites depending on different sugars it takes. Independent of the different sugars it takes, it tries to make the same set of 12 different precursor molecules. So you have a large set that is slowly narrowed down in a funnel-like manner. That pattern is called the bow tie pattern. Third question is presence of more amount of precursor molecule necessarily means higher level of biosynthesis that will be occurring naturally. So this was also discussed as a part of week two's problem solving session. So what one needs to understand is that just because a precursor molecule is available, that doesn't mean that the cell will utilize or synthesize more of the macromolecule that is needed. So the macromolecule that is needed is synthesized depending on the function. Only if a particular function is needed, that macromolecule is being synthesized. And only if that macromolecule is being synthesized, then more of the precursor will be utilized for the synthesis. So that way, the answer here is false. Based on macromolecular constituents of cellular materials, choose the option that correctly represents the arrangement of macromolecules in order of most abundant to least abundant. So uh, for this, if we look at the lecture, in one of the lectures, the professor had given a distribution of how much of proteins is present in the cell, how much of RNA, DNA, and peptidoglycan is present in the cell. So in general, within a cell, the functions are majorly carried out by proteins. Or okay, I'll explain it in a different manner. So within a cell, usually the DNA content, it is all combined and present as the genome or the chromosome. So it doesn't change with time. What changes with time is rather the other components that are derived from the DNA, which is basically the RNA. So when DNA is being transcribed, we get different RNA. And the number of RNA that is being produced within the cell, it is both being produced and decayed. So that depends on the kind of expression that it is needed. And from one RNA with translational apparatus that is present in the cell, multiple copies of proteins, hundreds and thousands of copies of proteins can be created. So that way, the amount of protein within the cell is always greater than the amount of RNA, and the amount of RNA within the cell is always greater than the amount of DNA within the cell. And peptidoglycan is a part of the cell's outer membrane. So this also has a constant composition that doesn't change with time, uh, although it is being continuously replenished, the amount still does not exceed as any of the other components that are described previously. Question 5. Uh, Precursor metabolites used for the production of building blocks of cell wall in E. coli is, it is given as fructose 6 phosphate, but as we had this discussion with Surender, that because the cell wall is majorly comprised of NAG and NAM, and because it has the N-acetyl glucosamine, which can be created from glucose, aminated glucose. Uh, it could be option A, but I will check with the team and I can answer further. Question 6. Which of the following is not true for nutrient uptake by the cell? Option A. Non-specificity of the uptake process. Option B. Uptake of the molecule down or against the concentration gradient. Option C. Nutrient molecules cannot pass through any cellular membrane. So, to answer this question, we can think about when a cell takes up a nutrient. The cell does not randomly takes up a nutrient. Only if there is any function that the cell has to perform, and if it needs energy, it takes up the nutrient. So, that way, uh, option A, which is non-specificity of the uptake process, is not true for nutrient uptake. What is the redox nature of the anabolic reaction and catabolic reaction, respectively? Anabolic reaction, catabolic reaction. It is given as a reduction reaction and oxidation reaction. So, if we look at catabolic reactions, these are basically converting sugars into acids. So, sugars have hydroxyl groups, which are alcohols, but acids are oxidated forms of alcohol. So, when alcohols are oxidized, they form aldehydes or ketones. When aldehydes or ketones are oxidized, they get converted into acids, pyruvate for instance. So, catabolic reactions are oxidation reactions, whereas anabolic reactions that give energy, here We also, in both cases, we observe the involvement of uh, redox cofactors. And since catabolic reaction is involved with oxidation reaction, we can go with answer option B. Question 8. Suppose you're working with a cell line that is growing in a medium with P32 and galactose as carbon source. The cells also have a mutation in a gene whose product is responsible for the interconversion of high energy intermediates that connect the layer pathway with PPP and EMP. This gene product can only function in a forward direction. What will be the correct sequence of the position of radiolabel phosphate addition? Okay, I need to check the pathway to be able to answer this question, to be able to answer the three of these questions. Uh, yeah, Surendra. So basically, we tried to cover what we had planned to cover in this session. If you have any further questions, we can discuss that, or we can end the session today now.
There is no query from my side.
Okay, thank you. Thank you for attending.
But
Yeah,
the correct answer for my query will be rectified in the assignments or not?
I don't know. I need to
Okay,
ask the team. I am not sure. I'll check and I
okay.
can tell you that.
Is there any specific email where I can put my message emails through email?
I don't know. I can maybe
Okay,
write to the professor. So we are not directly connected with the question making or the assignment team. We are supposed
okay,
to interact and have a problem solving sessions. So our our roles end there. But I will check if that's the right answer or if that's the wrong answer, and I can try to see what can be done. I'll do that.
okay.
Yeah.
Thank you.
Thank you.